Hello, this is Dr. Kodev, and this is Human Anatomy 260, Martini, Chapter 20, the cardiovascular system, blood. Our cardiovascular system, so cardio means heart. Okay, so cardio, whoa, means heart. Cardiovascular system functions as a system to transport numerous substances throughout the body. So this is how our blood is going to give us our nutrients, oxygen, carbon dioxide is going to cruise through here, different hormones, ions, metabolic waste, white blood cells for our immune system. Our blood is really the ultimate connective tissue, connecting every part of our body. And it consists of two components, plasma, the liquid matrix of blood, and formed elements. And I have a little video for you on this and a little note packet for you to help illustrate this as well. So we have our plasma, the liquid matrix of blood, and the formed elements, which is blood cells, cell fragments that are suspended in the plasma. And it's going to include this whole blood. So males typically will have about 4 to 6 liters of blood, females about 4 to 5 liters, pregnant females even more than that, hypovolemic, normovolemic, and hypervolemic. So hypo means below. So these are below normal blood volumes. Normal would be normal. Hyper is excessive. So this would be excessive blood volume. Important to note, the pH of blood is about 7.3 to 7.45. This is tightly controlled through a couple of systems, namely your respiratory system and your kidneys. The temperature of blood is about 100 to 100.5. Plasma makes up about 55% of the volume of whole blood, consists about 92% of water, about 7% protein, being albumin, globulin, fibrinogen, and regulatory proteins. About 1% would be other solutes like electrolytes, organic nutrients, and organic waste. Our plasma proteins, there's 3 major classes of protein in the blood, and they are going to be albumin, globulin, and fibrinogen. So we have about 60% albumin, which contributes to the osmotic pressure, transports fatty acids and steroids, and this is the smallest of the proteins. Now, 35% are going to be globulins. Now, globulin, we'll see the same word here in our immunoglobulins or antibodies. So think immune system here. They act as transport proteins and transport ions and hormones for us. About 4% is going to be fibrinogen. They're involved in blood clots, and they're the largest. Our formed elements makes up about 45% of whole blood. Our platelets, which are responsible for clotting, is really, really small. Now we see these leukocytes. Leukocytes are also known as white blood cells. So small amount of that whole blood as well. And when we subdivide these leukocytes or these white blood cells out into neutrophils, eosinophils, basophils, lymphocytes, and monocytes. Neutrophil is neutral, is how I think of that, neutral, and he's the most common. About 50 to 70% of white blood cells are going to be neutrophils. Eosinophils, about 2 to 4%. Basophils, less than 1%. Lymphocytes, 20 to 30%. Monocytes, 2 to 8%. And then our red blood cells, erythro is red, remember? So red cells, red blood cells, 99.9% of our whole blood. So this is that 0.1%. And then of that 0.1% is this here. Okay. So our red blood cells were erythrocytes. We see hematocrit on blood work, and hematocrit is the ratio of volume of red cells to the volume of whole blood. And this is checking a lot of times for anemia. A in front of a word or an in front of a word is without. Anemia is condition of the blood. So this is saying we don't have blood. We do have blood. It's just anemia. Okay? Polycythemia would be an elevated hematocrit. And here is a typical red blood cell. So you can see it's biconcave. So on both sides, it has this concave or dip in the middle. Okay? And you can see narrower here and wider here, and we'll look at that. So it's a biconcave disc, thin central region, measures about 7.7 microns in diameter. It lacks organelles, the little tiny organs. It also lacks a nucleus, or what we would call anucleated. The lifespan of a red blood cell. So since red blood cells lack a nucleus and all the organelles, it only has a lifespan of about 128 days, which is really good news because you can have a whole new red blood cell supply in that 120 days. Okay, so a lot of times when someone has a glycosylated hemoglobin or too much sugar, to put it plainly, on the red blood cell, it takes about 120 days to heal from that. And we're not stuck with that permanently, with major changes happening if possible, right? Not everything is possible depending on what the condition is. Significance of a lack of a nucleus. So this allows the cell to be flexible as it travels through the circulatory system. See how much more flexible this is than rigid? It allows for more room for hemoglobin, so we can carry more oxygen. The significance of a lack of a mitochondria. So that is the powerhouse of the cell, right? But mitochondria use oxygen to manufacture ATP. So we don't want that mitochondria in there using up all of our oxygen. So that oxygen can be transported to other tissues. Red blood cells and hemoglobin. So a developing erythrocyte red blood cell loses its nucleus and organelles. A mature red blood cell or erythrocyte is mainly just a cell membrane surrounding water and protein. Water accounts for about 66% of the red blood cell's volume. The protein accounts for about 33% of the red blood cell's volume, which is greater than, right? We'll see 95% and greater as hemoglobin. Hemoglobin is responsible for transporting our oxygen and our carbon dioxide. You can see the four little heme groups here on our hemoglobin. Now, blood types. Blood types are determined by the antigens on the surface of the red blood cell. These antigens are also known as agglutinogens. These agglutinogens are either glycoproteins or glycolipids, so sugar and protein or sugar and fat. There are three major types. So blood agglutinogen A, B, and D, and agglutinogen RH as D. White blood cells are leukocytes. There are two major classes of leukocytes, and we're going to see them as granulocytes and agranulocytes, and a total of 5 major types of these leukocytes. So we're going to see neutrophil, eosinophil, basophil, monocyte, and lymphocytes again. Let's look here. So there's about 6,000 to 9,000 leukocytes per microliter of blood. A total white blood cell count is performed on an instrument called a heme blood cell site, and meter is just for measure. Hemocytometer. A low count is called leukopenia. Penia is deficient when we see that. An elevated cell count is going to be leukocytosis. Osis is usually just abnormal condition of. Okay, so this is an abnormal condition of specifically elevated. A differential count is performed to determine which of the leukocytes is in excess or deficient. So we'll do a differential. So here's the differential of the granulocytes. So we see our neutrophils, normal range about 50 to 70%. These granules, right, contain chemicals to kill bacteria. Typically, this is the first on the scene. This is your first responder, and it's going to respond to our bacteria. very active phagocytic cell, so eat, okay? So it eats the cells like a Pac-Man. And the nucleus is multi-lobed. The eosinophils normal range is about 2 to 4%. The granules release chemicals that reduce inflammation. It attacks a foreign substance that has reacted with our circulating antibodies, like an allergic reaction or parasites. So eosinophils are really just for those allergies and parasites. And in countries that have more parasites, we see less allergies. And in countries where we see less parasites, we see more allergies. So interesting fact there. They are also bilobed. Basophils. Now, these granules release histamine and heparin. And histamine, think about when you take an antihistamine because you're all red, swollen, and itchy. Okay? And then heparin is going to do the opposite of blood clotting. So it's going to thin the blood. Okay? So histamine dilates our blood vessels to get more stuff, healing stuff to the site. Okay? It's going to be red, hot, itchy. Okay? And then heparin preventing abnormal blood clotting. And the nucleus is usually hidden due to all the granules. Monocytes, the normal range is about 2 to 8%. They're large eating cells, phagocytic cells, including large eaters, macrophages, Langerhans, and microglials. Lymphocytes normally range is 20 to 30%. It's responsible for specific immunity. Cells differentiate to form our T cells and our B cells and our natural killer cells. The nucleus is typically large and round, leaving a small halo around the entire nucleus or part of it. Our platelets are derived from megakaryocytes. So mega is big, kary is a nucleus, and then cell, okay? Megakaryocytes. And they break or fragment that looks like a big plate, right? Looks like a big plate, and then it looks like you drop the plate and it fragments and it forms bits and pieces of membrane enclosed packets of chemicals. And their main chemical is a platelet thromboplastin factor. So this is blood clotting. About 350,000 per microliter of blood, formerly called thrombocytes. Okay, platelets, thrombocytes. So we'll still call it thrombocytopenia when there's a lower than normal amount of platelets. And then a higher amount we're going to call thrombocytosis. Platelet function involved in blood clotting for hemostasis so we don't bleed out. Releases chemicals to initiate the clotting process. Clump together to form a platelet plug and contain actin and myosin that function to contract the clot. Now, actin and myosin might sound familiar from the muscle chapter. Actin is that thin filament, myosin is that thick filament. All right, so you have this available to you in slide form here that you can draw on, right? Or you can download it as a separate note packet. So I'm going to play you a little video here, and that will take us to the end of this chapter.
Here we'll learn the composition and function of blood. To begin, let's start a table to categorize the components of blood. To note that blood is a connective tissue suspended in plasma. To note the two key components of blood: plasma, a straw-colored liquid, and formed elements, which are cells and cell fragments dispersed in plasma. Their even dispersal relies on good blood flow. As a clinical correlation, denote that poor blood flow can cause blood fragments to clump in the veins and form deep venous thrombosis known as DVT, which is a potentially life-threatening condition, especially if it results in blockage of the pulmonary arteries. To better understand the components of blood, let's pretend to use a centrifuge to separate plasma and formed elements. Draw a test tube filled with blood that's evenly dispersed. indicate that we place this test tube in a centrifuge, which rapidly spins it. We use an anticoagulant to prevent clumping. Next, draw another test tube with both red sediment of formed elements at the bottom, it's the most dense substance, and straw-colored plasma on top, it's the least dense. Indicate that the separated blood comprises 45% formed elements and 55% plasma, an almost 50-50 mixture. Now let's examine the key constituents of the plasma and the formed elements. Denote that the plasma comprises the following: water, which functions as a transport medium, and proteins, which are the most abundant organic constituents in the plasma, as well as electrolytes, which are dissolved ions, and metabolic nutrients and waste, which include the respiratory gases oxygen and carbon dioxide, and also hormones, which are numerous and bear many different functions. Next, denote that the formed elements comprise the following: erythrocytes, also known as red blood cells, which transport oxygen and carbon dioxide; leukocytes, also known as white blood cells, which protect the body from foreign organisms; and platelets, also known as thrombocytes, which are fragments of large cells. Now we're ready to learn their specific functions. Again, let's start with the plasma. Illustrate that the plasma in our test tube contains 90% water, which importantly maintains a high capacity to hold heat. So write that water functions as a transport medium and is able to absorb and distribute heat throughout the body. Plasma buffers blood temperature while it distributes heat from the tissues. Next, show that the remaining 10% of plasma includes proteins, electrolytes, metabolic nutrients, metabolic waste products, and hormones. Of this 10%, plasma proteins constitute the greatest portion, so let's examine them first. Write that collectively plasma proteins buffer blood pH, maintain an osmotic balance between the interstitial fluid and the blood, and give blood its viscosity. Let's focus for a second on osmotic pressure. Because plasma proteins are the largest of the plasma's constituents, they rarely pass through capillary pores to enter the interstitial space, but instead remain within the blood. Thus, they produce the osmotic pressure that helps drive solute exchange in the capillary beds. Now let's learn some specific functions particular to each plasma protein beyond their collective functions. Show that specific plasma proteins include albumin, which is the most abundant plasma protein. Write that its relative abundance makes it the principal contributor to osmotic pressure. Albumin is made in the liver, and thus in liver failure patients, low albumin results in abdominal swelling or edema due to poor osmotic pressure. Immunoglobulins, also known as antibodies, write that they function in immune defense. Fibrinogens, which help plug damaged blood vessels, write that they are one of many clotting factors. As a clinical correlation, denote that hemophilia occurs when a genetic mutation causes a deficiency in one of these factors. Patients with hemophilia bleed excessively, even from minor cuts and bruises. Now that we've learned the major plasma proteins, let's finish characterizing the rest of the plasma's constituents. Write that similar to plasma proteins, plasma electrolytes buffer blood pH and help maintain the blood's osmotic balance. Write that they also regulate membrane permeability. These ions, which the kidneys closely regulate, include sodium, potassium, magnesium, and many others. They are essential to muscle and nervous system function. Lastly, let's note the presence of metabolic nutrients and waste in plasma. Metabolic nutrients, such as glucose, fatty acids, and vitamins, are absorbed from the digestive tract. Metabolic waste, such as urea and uric acid, are processed by the kidney and discussed in detail elsewhere. Now that we've characterized the plasma, let's illustrate the formed elements. Show that erythrocytes constitute 98 to 99% of the formed elements in our test tube. Erythrocytes lack nuclei in most other organelles. Write that erythrocytes transport oxygen and carbon dioxide in the body. They use hemoglobin for this transport. Next, illustrate that leukocytes and platelets constitute the remaining 1 to 2%. Platelets are the more abundant of the two. Show that leukocytes, also known as white blood cells, can be categorized into granulocytes and agranulocytes. Think of the granule and granulocytes as granules, which describes the appearance of small dark spots that appear when granulocytes are stained in a lab. Granules function in the immune system. Agranulocytes have inconspicuous or no granules. Illustrate that granulocytes include the following: basophils, which play a role in the inflammatory response and maintain a large concentration of intracellular granules; eosinophils, which multiply during parasitic infection and allergic reactions; neutrophils, which are the most abundant of the three and engulf bacteria. Illustrate that agranulocytes include the following: monocytes, which are large cells with a kidney-shaped nucleus that leave the bloodstream to engulf bacteria; lymphocytes, which are categorized into B and T cells and differentiate in the bone marrow and the thymus respectively. Leukocytes play a major role in the immune response, and each subtype contributes to the body's defense system in a unique yet coordinated way. Finally, write that platelets, like fibrinogens, function in blood clotting, which is also known as hemo for bleeding, stasis for stopping. Platelets lack nuclei because they are fragments of megakaryocytes, which are large cells in the bone marrow. This concludes our diagram.
Great job. Make sure that you rewind, pause, play as many times as you need in order to study this the best for you. And this is just a study tool for you. You do not have to turn this in anywhere. This is not for points, just a little study help for you. All right, that concludes our chapter, and I'll see you at the next chapter. Thank you.